Hello, my name is Michel Lapointe, and I'm going to present to you the research that I'm doing for my PhD in biomolecular sciences at Laurentian University. But first, I want to point out my affiliations with the Nuclear Innovation Institute and Bruce Power. I'd also like to point out that this research is partially funded by the Nuclear Innovation Institute, Bruce Power, and the Natural Sciences and Engineering Research Council.
Moving on, the title of my poster is Yeast Anhydrobiosis and Radiation Resistance. We're using yeast as a model organism for this project because we want to better understand how radiation damage is repaired, and yeast are quite special in that they're capable of entering a state of suspended animation called anhydrobiosis. This just means that yeast can survive complete desiccation, or drying, for an extended period of time. While fully desiccated, all metabolic activities within the cell are ceased, which includes DNA repair. As such, it is hypothesized that DNA damage from the environmental background radiation would accumulate while in the desiccated state. By comparing the long-term desiccation survival of a strain of yeast that is incapable of repairing DNA damage from radiation with the survival of normal wild-type strains, we can infer the environmental radiation dose rate. This technique of using desiccated yeast as a living radiation dosimeter is currently being used by our collaborators at NASA Ames Research in their BioSentinel mission. They're using these biological dosimeters in deep space to help quantify the dose rates in this region to better inform the risks of space travel. We, however, are looking at quite the opposite situation. We're sending our yeast dosimeters into the ultra-low radiation dose rate environment inside SNOLAB, the Nobel Prize-winning physics laboratory here in Sudbury. In doing so, we hope to see more similar survival over time between the radiation-sensitive yeast and the wild-type ones, when compared to normal background stored control samples. If this is indeed what we observe, we would be confirming that DNA damage accumulates while desiccated, and we'd be validating the use of desiccated yeast as living radiation dosimeters.